Good morning, everyone, and welcome. Thank you for being here today. I am pleased to introduce our presentation on analytical method verification. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we'll explore the key aspects of verifying analytical methods, ensuring accuracy, precision, and reliability. Over the next few slides, we'll cover the main steps involved, best practices, and important considerations. So, let's get started and dive into the fundamentals of analytical method verification. Welcome to today's discussion on analytical methods. As we know, these methods are crucial in ensuring the quality of pharmaceuticals. They help us confirm the identity of the substance, verify its purity, and assess its potency. Each of these factors is vital to guarantee that a medication is safe and effective for patients. Now, let's clarify two important terms: verification and validation. Validation is about developing a new analytical method from scratch. It involves demonstrating that this new method is suitable for its intended purpose, ensuring accuracy and reliability. Verification, on the other hand, is about confirming that an existing compendial method, like those from USB, IP, or EP, is still appropriate when applied in our specific lab environment. It's a way to ensure that the method works well under our actual conditions. This step becomes especially important when we adopt pharmacopoeial methods. We need to verify that these methods perform as expected here, so the results we get are trustworthy. Overall, both verification and validation are key to maintaining high quality standards and ensuring compliance with regulatory expectations. So let's talk about when verification is actually required. First, it's important to understand that verification is needed when using certain types of methods. This includes compendial methods, such as those found in USB, IP, or pH. These are standardized methods that are widely accepted and have specific requirements. Additionally, if you transfer methods from one lab or system to another, verification becomes necessary to ensure consistency and reliability. Now, there are specific changes that trigger the need for verification. These include changes in equipment, because different equipment can affect the testing process. Changes in the analyst, since different analysts might interpret or perform tests differently. And changes in laboratory conditions, such as temperature, humidity, or other environmental factors that can influence results. It's also key to remember that if you're working with fully validated in-house methods, verification isn't usually required, unless you've transferred those methods to a new system or location. So, in those cases, the validation already covers the method's robustness, and additional verification isn't necessary. Understanding these points helps ensure we maintain compliance and the integrity of our testing processes. When we talk about parameters for verification, it really depends on the method type we're using. Different methods require us to focus on specific key points. For example, some methods might prioritize accuracy, while others emphasize speed or robustness. It's crucial to understand what parameters are most relevant to ensure the verification process is thorough and reliable. By tailoring these parameters to the method, we can better assess the performance and validity of our results. This connection is essential to maintaining the overall integrity of our verification process. Let's talk about specificity now. This is all about how well our method can measure the analyte even when other substances are present. These substances might include impurities, degradants, or excipients, all of which can potentially interfere with our measurement. For example, in an API assay using HPLC, we want to be sure there's no interference at the analyte's retention time. This means our method should be able to distinguish the analyte clearly from any other substances that might be there. The key point here is that any interference from these other compounds should be negligible, ideally less than 0.1%. This ensures that our results are accurate and reliable, and it highlights the importance of specificity in analytical methods, especially when we're analyzing complex mixtures. So, in the context of the overall presentation, specificity is crucial because it guarantees that our method is truly measuring what we intend to, without being confused by other components. let's take a closer look at the accuracy of our recovery study. Here, we're focusing on how precise and reliable our recovery process is. Essentially, we're assessing whether our methods can consistently restore the system or data to its original state. This is a critical component because accuracy directly impacts trust and effectiveness. If our recovery isn't accurate, even small errors can lead to bigger issues down the line. Throughout this study, we've been measuring how close the recovered outcome is to the original, and the results are promising. They show that our approach maintains a high level of precision, which supports the overall goal of ensuring dependability and integrity in our system. So, in the context of the entire presentation, this accuracy underscores that our recovery process isn't just functional, it's reliably precise, which is essential for building confidence in the system's resilience. Let's talk about precision. This concept is all about accuracy and consistency in our measurements or predictions. In many fields, especially in data analysis and machine learning, precision helps us understand how many of our positive results are actually correct. Think of it as a way to measure how trustworthy our positive findings are. The higher the precision, the fewer false positives we have, which is crucial when the cost of false alarms is high. For example, in medical testing, high precision means that when a test says someone has a condition, it's very likely they actually do. Connecting this to our overall theme, focusing on precision ensures we're making reliable decisions based on our data. So, by emphasizing precision, we can improve the quality of our outcomes and build greater confidence in our results. Let's talk about linearity. This is all about the ability to obtain a proportional response, meaning that as you change the concentration, the response should change in a consistent, predictable way. To evaluate this, we typically look at 5 different concentration levels, which might range from 50% to 150%, for example. The key idea here is that if your method is linear, doubling the concentration should roughly double the response, tripling it should triple the response, and so on. This is important because it ensures that our measurements are reliable across the range we're interested in. When we establish linearity, we're confirming that our assay can accurately reflect changes in concentration, which is essential for precise quantification. So, understanding and verifying linearity helps us trust that our results are proportional and valid throughout the entire analytical range. Let's talk about the limit of detection, or LOD, and the limit of quantitation, or LOQ. These are 2 critical concepts in analytical methods. The LOD is essentially the smallest amount of a substance that we can reliably detect, but not necessarily measure precisely. It's like the faintest signal we can pick up confidently. On the other hand, the LOQ is the lowest concentration at which we can quantify the substance with an acceptable level of accuracy and precision. So, while LOD tells us if something is present, LOQ tells us how much is there, with confidence. Understanding these limits is key because they define the sensitivity of our analytical method. They help us determine whether our method is suitable for detecting and measuring trace levels of substances, which is especially important in fields like environmental analysis, food safety, and pharmaceuticals. Connecting this to the overall theme, establishing clear LOD and LOQ values ensures the reliability of our results. It also helps us set appropriate detection thresholds and compliance standards. So, in essence, mastering these concepts ensures our analytical methods are both sensitive and accurate, which is the foundation of trustworthy data. Let's talk about robustness. This is all about how well our method can hold up when small changes happen in the process. For example, if the flow rate varies by plus or minus 0.1 ml per minute, or the pH shifts by plus or minus 0.2, or if there's a slight temperature fluctuation. These are minor variations, but they can still impact our results. So, it's important to evaluate how these small changes affect key parameters like retention time, peak area, and resolution. Ensuring robustness means our method remains reliable and consistent, even when things aren't perfectly controlled. This is crucial for producing trustworthy data across different labs and conditions, and it ties back to our overall goal of developing a method that's both accurate and dependable despite small variations. Now let's talk about system suitability testing, or SSD, which is a crucial step before we begin any analysis. Its main purpose is to ensure that our system is performing optimally and reliably. When we perform SSD, we look at several key parameters. First is the %RSD, or relative standard deviation, which should typically be less than or equal to 2%. This tells us about the precision of our system. Next, we evaluate the tailing factor, which should be less than or equal to 2, to assess how well the system is separating peaks without excessive tailing. Lastly, we consider the number of theoretical plates, which should be at least 2,000, indicating good column efficiency. Ensuring these parameters meet their criteria helps us confirm that our system is suitable for accurate and reproducible analysis. Remember, SSD is all about validating that the system is performing consistently before trusting the results of our samples. Now, let's take a closer look at our case study involving the API assay using HPLC. Here, we're focusing on the drug paracetamol API, and the method employed is the USP assay, which is a standard approach for determining the potency of active pharmaceutical ingredients. We performed several key verification steps to validate this method. First, accuracy, which tells us how close our measurements are to the true value, was recorded at 99.2%. This high accuracy indicates that our method reliably measures the actual amount of paracetamol API present. Next is precision, which assesses the consistency of our results. The %RSD, or relative standard deviation, was only 0.8%. That's a very low variation, showing our method produces reproducible results across multiple tests. Then we have linearity, which is crucial for ensuring our method provides proportional responses across a range of concentrations. The R-squared value was 0.9995, very close to 1, confirming excellent linearity. Putting all these verification points together, the conclusion is clear. This method is suitable for routine use. It's reliable, accurate, and precise, which are all essential qualities for an analytical method in pharmaceutical quality control. This case study reinforces the overall message that thorough validation ensures our analytical methods are robust and fit for purpose, ultimately supporting safe and effective drug manufacturing. Let's take a look at this case study focusing on the finished product, specifically the tablet formulation. Here, we conducted a dissolution test to evaluate how the active ingredient releases from the tablet over time. The results were quite promising, with a release percentage ranging from 98 to 101%. This indicates that nearly all of the active substance is being released as expected, which is a great sign of the formulation's effectiveness. Additionally, the %RSD, or relative standard deviation, was only 1.2%, demonstrating excellent consistency and reproducibility in our testing. These results are critical because they show that the product performs reliably within the desired parameters. As a result, we've verified that this formulation is suitable for routine quality control use, ensuring that every batch meets the same high standards. This example highlights how thorough testing and solid data underpin confidence in our product's quality and performance. Let's wrap up with the conclusion. As we've seen, verifying our analytical methods is crucial to ensure the reliability of compendial methods. This way, we can confidently trust the results we generate. It also helps us stay compliant with global regulations, which is essential in today's international pharmaceutical landscape. I want to highlight the key guidelines that govern this process, specifically ICH Q2, R2, and USB 1226. These provide the standards and best practices we need to follow to validate our methods properly. This verification process is especially critical when it comes to testing active pharmaceutical ingredients and releasing finished products. Accurate and validated methods ensure that what we produce meets quality standards before reaching patients. Finally, I can't stress enough that method verification is essential for regulatory inspections and maintaining data integrity. It builds trust with regulators and ensures our data is solid and defensible. Overall, rigorous verification supports both compliance and quality in every step of our manufacturing process. For further support, please contact us. We're here to help you with any questions or additional information you may need. Don't hesitate to reach out. Your success is our priority. And with that, I want to thank you all for your time and attention today. It's been a pleasure sharing this information with you, and I look forward to connecting further. Thank you again.